I heard that. No, really, I did hear that because we're talking about hearing. Holes, chapter 12, the ear. It's the organ of hearing, 3 sections. External, middle, and inner. I can't really point to the middle and inner because they're inside. Out here we have the auricle. This is the satellite dish, if you like, on certain people, like my dad. Yeah, I know, I'm screwed when I get older because your ear keeps growing as you get older, bigger and bigger, so does your nose. I'm screwed in both nose and ear categories because they're going to keep getting bigger. The good news is I have an evolutionary advantage. I will be able to hear better than most people because this satellite dish called the auricle will funnel sound into my middle ear. On the way to the middle ear, it's going to pass through the external auditory meatus, which is lined with ceruminous glands. You might remember that these ceruminous glands make earwax, and I have a fair amount, just like everybody else. And the sound travels through there and meets with the tympanic membrane that you see right here, which is a little flap of tissue, which will move like this when sound, my finger is sound, and the sound moves it and it causes it to flap. Okay, and we'll get to why in a second. The tympanic membrane vibrates in response to sound waves. Fine, great. So then inside we have the tympanic cavity, an air-filled space in the temporal bone. You'll see why that's important later when we talk about the round window. You'll remember hearing the round window. It's round because it's round. And the auditory ossicles. These are found in the middle ear. These are the smallest bones in your body. Although you can theoretically break them, nobody really does. These include the malleus, the incus, and the stapes, or stapes, which have other common names like hammer, anvil, and stirrup, but we call them malleus, incus, and stapes here. The first one that's attached to the tympanic membrane excuse me, is the malleus. The malleus is attached to the incus, and the incus is attached to the stapes, and the stapes is attached to the oval window, which we'll show you in just a bit. You can't really see it here, but the oval window is an actual oval flap of tissue that when the malleus vibrates, and then the incus vibrates, and then the stapes vibrates, well, the oval window vibrates, and that causes the fluid inside the inner ear to vibrate as well. And that's a dance you can now do since you've learned malleus, incus, stapes, oval window. Auditory tube. So you do have a connection. This is your eustachian tube, also called the auditory tube. You can notice here, it's this tube here. A lot of students will confuse it with the external acoustic meatus. That's not what we're talking about. The auditory tube or eustachian tube connects to your throat, your mouth. And this is how you get ear infections, people. Ear infections are not caused from water being stuck in your ear because your tympanic membrane is there. They're caused from internal infections in your throat that climb up through your auditory tube or eustachian tube. And in fact, its purpose is to help maintain equal pressure on both sides of the tympanic membrane. You've all felt this when you've been in an airplane and your ears start to feel like they're going to pop, or you feel that buildup of pressure. So basically what you can imagine is if this is the outside of my tympanic membrane and this is the inside, when pressure outside builds up, when I go to higher elevations, the pressure outside is lower than the pressure inside my ear, so that forces the tympanic membrane to bend out like this, and it stiffens and hardens. And as a result, you can't cause any vibration from sound coming in because there's a higher pressure pushing on the back here. And so what you'll do is you swallow, and you open up, you'll see here in this little animation, increased pressure from the outside, such as when you're going down versus the inside, or vice versa. You'll equalize the pressure out by swallowing and opening up when you swallow this tube, bringing in air from the outside back in to equalize the pressure on both sides of this membrane. And now it's able to flap again. It's not all tense and taut and not able to flap. So that's kind of how or the purpose of your eustachian tube, and swallowing opens up the valve, such as when you're in an airplane or diving, that kind of thing. Now we get to the inner ear, the complicated part. Okay, this is where hearing really takes place and happens. It's a complex system of labyrinths, all kinds of tubes and things going different ways, filled with different fluids. We have the osseous labyrinth. This is bony. Osseous means bone. And it's in the temporal bone, of course, and it's filled with perilymph, all these different kinds of lymph. Perilymph is the one on the outside. You then have the membranous labyrinth, which is represented here in the cochlea by the purple that you can see here. And inside that purple, you have the membranous labyrinth, and it's inside the osseous labyrinth, and it's filled with endolymph. Endo is inside. So now let's look more deeply in. You can see here the bony labyrinth on the outside. Okay. Then you have perilymph inside that, and then within that you have the membranous labyrinth, as we mentioned, and inside there you have endolymph. So this is a nice cross-section to explain that. The three parts of the labyrinth include the cochlea, which we're going to function on here for this part. The next part we'll talk about the semicircular canals and the vestibule, because those are equilibrium and movement. But the cochlea is your hearing organ on the inside. Okay? The cochlea is made up of two main sections. We have the scala vestibuli, and the scala vestibuli is found here on this outer region. It's directly attached to the oval window. Here you see the stapes attached to the oval window. And the oval window is a flap of tissue that's going to flap and is attached to the scala vestibuli. And when this oval window moves like this, it's going to cause the fluid inside the scala vestibuli, again, perilymph, to move as well. Okay? And when the perilymph moves, you'll see it moves the fluid down here, and it's going to cause, you'll see the fluid inside the membranous labyrinth to also move, but we'll get there in just a second. At the bottom here, we have the scala tympani, and this is connected at the very end to the round window. Again, you heard me say that before. This is the lower compartment on the other side of the membranous labyrinth. There is a little passageway here, this helicotrema, although not an important structure, where fluid passes through, but the vibrations are not going to really pass through the fluid in this way. They're going to pass vertically, I was going to point to the screen, vertically down through the membranous labyrinth and then into the scala tympani. So a little bit weird to think about. Think of this being the scala vestibuli up here with perilymph, and the perilymph moves, and that causes the fluid, the endolymph, and the membranes that are the membranous labyrinth to move like this. Okay, and then down here we have the scala tympani, and that causes the perilymph that's in the scala tympani to now move as well. And we'll talk about why that is in just a second. We'll focus in now on this region in here, the cochlear duct. Okay, the cochlear duct is a portion of the membranous labyrinth. Here you see the membranous labyrinth. Okay, it's that purplish region again. On the top, once again, scala vestibuli, on the bottom, scala tympani. And the vestibular membrane that you see here is a membrane that separates the scala vestibuli and the perilymph with the cochlear duct. Okay, and the endolymph that's inside the cochlear duct. We then have the basilar membrane, and it's called basilar membrane just like we saw in epithelial cells, because these hair cells are actually modified epithelial cells that actually act very similar to neurons, but they are epithelial cells. So they are attached to a basilar membrane, and this basilar membrane will also vibrate when the endolymph vibrates, and that you'll see is going to cause these hair cells to move or vibrate. And when the basilar membrane vibrates, it also causes the perilymph here in the scala tympani to also vibrate or to move. And then the round window helps to absorb all those vibrations in the end, to stop them so that they don't flow backwards. Okay, now inside the cochlear duct is what we call the organ of Corti. Now the organ of Corti, or Corti, is these hair cells. Okay, it's a group of hearing receptor cells on the basilar membrane, so they're attached. And each different hair cell responds to a different frequency of sound, and that's because each frequency of sound has a better ability to penetrate or to cause the fluid of the endolymph to move or to vibrate as it travels down the vestibular, the cochlear duct. So can I show you? Yeah, so what you see here is all along the length, starting from here, going down, there are hair receptor cells. And as you go around, starting from the bottom here, the further you get, the higher the frequency sound those hair cells respond to. So higher frequency sounds are going to travel, be shallower depths, smaller wavelength, and more often, so they travel more easily through this thick fluid. And so down here you have the receptor cells for high frequency, and down here you have the receptor cells for lower frequency. So different frequencies of vibration move different parts of the basilar membrane, and that causes certain hair receptor cells to bend and to move. And when they do that, that you can see here, here's these hair cells attached to the basilar membrane. When these hair cells shift, what happens is calcium enters the hair cell, just like we saw with the axon terminals with neurotransmitter being released. And there's neurotransmitter inside those hair cells, and that neurotransmitter gets released because of the influx of calcium. And that neurotransmitter will activate or bind to neurons of the cochlear nerve. Okay? And an action potential will get generated, which will be sent to the medulla, and then from the medulla to the thalamus, and then from the thalamus over to the auditory cortex in the temporal lobe, so that you can process the sound in your cortex. So again, perilymph vibrates, vestibular membrane vibrates, causes the endolymph to vibrate inside the cochlear duct, causes the basilar membrane to vibrate. Attached to that are hair cells. These hair cells respond by calcium entering the hair cell, releases neurotransmitter onto the cochlear nerve, or cochlear neurons. Those cochlear neurons generate an action potential, runs to the medulla. From the medulla, it gets routed to the thalamus, which is going to direct things. Specifically for hearing, it routes it to the auditory cortex in the temporal lobe. There is some crossover, meaning that the sound coming in from the left ear goes to the right hemisphere and right ear to the left hemisphere, but mostly it stays same side. Hearing stays same side. It's very close. Here you see the real thing. One thing you'll notice here is this tectoral membrane. We do know that the tectoral membrane sits on top of these groups of hair cells, but we don't entirely know the function. We know it's important, but we don't really know why. The tectoral membrane, we do know moves and may be tied to the functioning of the action potential in the neuron, but we don't exactly know what the deal is with the tectorial membrane. This is showing you just the pathway that I mentioned. From hearing, you have the cochlear nerve sending information through the medulla down here, and then up into the thalamus, and then from the thalamus, it passes over to the auditory cortex. This is a summary of all the things. This is a great slide to know, one that I love to reference on tests, showing you step by step what happens from thing to thing to thing to thing. You should know this. And this is where we're going to pause and stop. Part 2 will be equilibrium.